Thank you, Peter. It's great to be here. It's my first trip to Australia, and it's been great so far. I want to tell you not just about the results that we've obtained on the question of emotion in the brain, but also a bit about how we do the research and where the answers come from. And I just want to start with an observation, which is that the human brain is it's part of natural biology, and it's a thing in progress. It's not. It's like a cake that's not quite cooked, and it will never be cooked, really. And it's there's a lot of things that are unfinished in many ways, and because we're always changing and becoming different organisms as we evolve. And one of the most important things that seems to be unfinished is the relationship between our emotions and our higher thought processes in our brains. So the systems involved in thinking, reasoning, planning, decision-making, and so forth are poorly connected with the regions that are involved in a lot of our more base kinds of emotions. So here's I guess I don't have a pointer up here, but here's the human brain, or a picture of the human brain from the outside. And what you see is all the areas of the brain labeled in terms of the lobes, and most of the back part of the brain is involved in the reception of sensory information, and then the more frontal areas are involved in the control of movement. But then the very front part is more involved in our higher thought processes, our ability to think and plan and make decisions and so forth. Now, I should just take a moment to say that I know this is a mixed audience, so for the neuroscientists in the crowd, I'm starting off, that's something for the lay people, and then I'll have something for the scientist later. So it's a tough line to walk when you do these kinds of lectures, but I hope I have a little bit of something for everyone. Now, the prefrontal cortex is part of the brain that greatly expanded with the evolution of mammals, with the evolution of mammals, and especially in primates and humans. And so we want to ask how this part of the brain is related to areas involved in emotion. Now, we're looking at the brain here kind of like a hot dog bun. So on the top part, we're looking at the outside, sort of the brown toasted part that you see when you look at a hot dog bun. And on the bottom panel, we see the hot dog bun with one side pulled apart. So if you've got the hot dog bun here and you pull away one part, you're looking at the middle where it's white and not toasted. And a lot of the systems involved in emotional responses are on this inside part of the hot dog bun, and the thinking parts of the brain more on the outside part. But it allows us to look at these connections. And one of the interesting things is that the prefrontal cortex, the lateral prefrontal cortex, where we have things like working memory and, again, these thinking, planning, decision-making parts of the brain, are not connected at all with some of the emotional regions that control things like fear responses and so forth. So that poses a problem right away. How can we control our emotions if our thinking part of the brain is not connected up with these emotional centers? And the problem is even more compounded because the emotional parts have massive connections with the cerebral cortex. So this gives us the situation where, while it's hard to control our emotions, it's very easy for our emotions to control our thoughts. Once you're afraid or anxious or depressed, it's very hard to just say, okay, I'm not going to be afraid anymore. There's kind of this domination of emotion once it takes over in your brain. So this separation of emotion and cognition has some consequences. Sometimes we express emotions like anger or other behaviors when something less volatile might be in our best interest. Or we may find it hard to withhold responses to things that are desirable, even though they may be potentially harmful to us. We make decisions, though they may seem carefully reasoned, might lack an appreciation for the emotional consequences for ourselves or others. And mental disorders in people are often reflecting these kinds of disconnections between emotions and thoughts. Sometimes we have emotions that can't be controlled by thoughts, and sometimes we have thoughts that are separated from emotions completely. So a better understanding of the emotional brain might help us solve some of these problems, even though we can't make the brain evolve any faster than it's evolving now. By understanding exactly how our brain is put together and how it's working helps us kind of come to terms with exactly what we can expect out of our brains and what we have to work around when we deal with them. So there are a variety of different ways you can approach the emotional brain. I'd just like to describe three. One is a kind of one-size-fits-all approach, and that was the goal of the limbic system theory, which started around the middle of the last century, around the 1950s. And it was a heavy theoretical concept in the sense that it was an idea about how emotions might be represented in the brain. Wasn't necessarily, I mean, it was based on a few observations, but it wasn't something that emerged out of research so much as an idea that was just sort of hinted at by a few observations. And it tried to explain all emotions in this one big concept. And that didn't work so well. There were a lot of problems with it, which I've written about, and you can look that up and read about it if you're interested. So there have been other approaches. One is sort of centered around something called basic emotions theory. And in this idea, we have different systems in the brain for different emotions. And sometimes it's said that we have roughly 6 basic emotions that are sort of biologically based and hardwired into the brain circuitry. These are expressed universally and recognized universally, supposedly in people around the world, and conserved to some extent in different species, especially closely related species. And when they're active, the organism, whether it's a human or an animal, feels the emotion that is active. So when the fear system is active, the person or the rat feels afraid. When the pleasure system is active, they feel pleasure. The anger system is active, they feel anger, sadness, and so forth. Now, I think this is a big problem if we were interested in really getting into the nitty-gritty of how the brain works, because there's no way for us to know what, if anything, another organism feels. It's hard enough to know what another person really feels. And so there's this thing called the problem of other minds in philosophy, where for all practical purposes, everyone in the room could be a zombie, a perfect zombie, and I wouldn't know it. I couldn't tell that any of you are real people if you were perfectly constructed as robots or zombies or however you want to think about it. But the problem gets even much worse when we try to understand what's going on in the minds of another animal. I'm not saying that rats don't feel anything. I'm saying whatever they feel, it's probably going to be very different from what we feel, because the higher parts of the brain that are ultimately responsible for making our conscious states are very different in rats and people. The prefrontal cortex, for example, is the rat doesn't have these lateral parts of the prefrontal cortex that the human and other primate brains have. So I think there are problems with this assumption that when you have, say, a fear system in the brain and it's active, you can expect that whatever species you're dealing with is going to feel something. So how do we get around this? So this other approach, which is the approach I've taken, is to look for one emotion process in the brain at a time. And I have process in big letters there because I'm not saying that there is a fear system in the brain that takes care of all kinds of fears, or an anger system that takes care of all kinds of angers. I think we can design experiments in animals that will allow us to ask questions about these things like fear processes. How does the brain detect danger and respond to danger? That doesn't explain everything we need to know about fear, but it allows us to get a handle on some basic mechanisms that underlie certain kinds of fear responses. So I'm not making any assumptions about, when I study rats, about what the rat feels. It may feel something, but that's not part of my research project. What I'm trying to do is understand how the brain detects and responds to significant events, and the particular class of significant events that I've focused on are those that are involved with the detection and response to danger. So here's we're going to talk about now how to study fear in animals. I've kind of hinted at this already, but what I've tried to do in my work is focus on mechanisms that, as I said, allow the organism to detect and respond to threats as opposed to feel afraid. We select a behavioral paradigm in which there's a discrete stimulus that elicits innate responses when the stimulus is presented. And it does this, it's important that the responses are innate because it means that every rat is going to respond in the same way. And this is very useful when you're trying to study the brain. If you can have a response that's so reliable, it greatly simplifies what you're trying to do. And that you have a stimulus, that's also very important. So if you've got a stimulus and a response, you're really much better off when you try to study the brain than if you are just studying some kind of state that the brain happens to be in. For example, there are models of depression, animal models of depression, but it's not so easy to simply turn it on and measure a response. You're looking at very complicated things, whereas in the study of these kinds of fear responses, you present the stimulus, the rat performs or expresses the response, and then your task is very simple. You just simply have to connect the dots in the brain between the stimulus and the response. And a lot of the pathways involved in sensory processing are already worked out for us. A lot of the pathways involved in controlling responses are worked out. So the front end and the back end of the brain is really already set. All we have to do is find the connections between those systems. And that's really what I've been trying to do for a while now. Now, since most fears that people experience are learned, we come into the world knowing how to be afraid, but what we learn about is what to be afraid of. We want our work to be relevant to the human brain as much as possible. So we try to study fears that are learned about. And again, we trace the circuit from the sensory systems through the motor systems, and that gives us a great advantage to have the stimulus and response as part of this. Then we can determine where in the circuitry the learning might take place. So we know that if the circuit involves 5 different areas interconnected in some kind of linear fashion, then the learning probably takes place somewhere within that circuitry. And if we can identify where the learning is beginning to happen in the brain, we can go into the cells and synapses that are learning and try to understand what kinds of genes and molecules are involved in gluing those synapses together to make the learning persist. So that's kind of the basic strategy that we've followed. And then once we have that information, we can verify, at least in the gross sense, that the circuitry is applicable to the human brain by performing very similar kinds of studies in humans. And my colleague Liz Phelps has been very good at pursuing some of these kinds of questions in the human brain, following the pattern of methods and results that we have in the rat brain. So, and then finally, as I've already said, but we don't make any assumption about all the connotations of the word fear. We're simply trying to understand a very simple kind of fear response, how the brain detects and responds to a discrete stimulus, not the fear of falling in love or the fear of being afraid or the fear of public speaking or anything like that. We're trying to these very simple kinds of fear mechanisms probably underlie some of these more complex fears to some extent. But we have to start small and build up rather than trying to explain everything all at once. Now, some of the steps in this, I didn't kind of invent this whole list of things myself. These are things that are kind of standard in our field. And a lot of it goes back to a paper in 1969 by Eric Kandel and Alden Spencer, where they mapped out a strategy for exploring how the brain learns in an invertebrate organism, in their case, a sea slug, the Aplysia. And so they said you've got to have a learned behavior, and then you trace the sensory to motor pathways, then you go in and find the cellular changes that underlie the learning, and then you identify the molecules. So this is really something, even though it's not always recognized or talked about so much, really goes back to this kind of Kandel and Spencer circuit approach to neuroplasticity and its molecular basis that's been around for a while now. And this was very influential when I was a young graduate student, and so this is the model that I've followed in my work. So let's get into some of the details here. We're studying rats. And the rat is placed in a small chamber, and it hears a sound. And if nothing happens, he begins to ignore the sound. But if the sound is paired with an electric shock, you only have to do this one time, and the shock is relatively mild. It's more like a kind of vibration, an irritating vibration, rather than you're not like frying the rats or anything like that. I mean, it's unpleasant, but I don't think it's like hugely painful to them. And we put our hands on the grids all the time just to verify everything is working properly. And so after you do this, again, you only have to do it one time, one tone and shock. Sometimes we do it 3 or 4 times, just depending on the experiment, but it's not a lot of exposure to the shock. Then you present the sound at some point later, and the rat will express this freezing posture that you see in the slide here. This is a species-typical posture that the rat will almost always express when it encounters a sudden danger, especially in a closed environment like this. And the rat will express, every rat will do it the same way, and the rat will do this to natural fears as well. So if he's exposed to fox odor or other things like that, he'll freeze, or if he's exposed to a cat or a predator, he'll freeze. And in terms of the learning, though, this rat will freeze the rest of his life when he hears that sound. You could condition him today and wait until the day before he's dying of old age, and he'll still perform this response. So it's a basically lifelong learning, and this is true in people as well. We develop fears, and then we carry them throughout life. You can go to therapy and treat the fear, but the fear can always be brought back by stress. So people who have, say, a fear of heights, and they're treated successfully through cognitive behavioral therapy, and they're doing fine, but then, say, the patient's mother dies and the fear comes back. This is a well-known recovery of fears and phobias by stress. So the bad news is fear is forever, but the good news is that if you know that, you can, you're not a bad person if the fear comes back. It's just the way the brain is. The nature's put these things in the brain so that fear is learned about very rapidly, because you don't have the opportunity to practice over and over again to get it right. If you're an animal in the wild, you better learn about it the first time, and you better remember it for a long time, because the things that are dangerous today are going to be dangerous tomorrow in all likelihood. So there are reasons why these things exist in the brain the way they do. It's not to make us have pathological fear. Pathological fear is a byproduct of these natural processes that are actually in the brain for good reasons. So in addition to freezing behavior, there's a lot of supporting physiology that goes on. Your blood pressure and heart rate go up, and respiration increases, stress hormones are released. Now again, I want to emphasize that these things happen not to make you feel stressed. They happen because they're part of the physiological response that the brain is taking care of to deal with the situation at hand. Because if you're a rat that's encountering a cat, you've got to get blood to the parts of your body that are going to be needed to escape or to fight the cat or whatever it is you have to do. And to do that, you've got to move the blood away from certain parts of the body and into the parts that need it. The blood, you can't create blood, you've got to get it there so that those parts have the metabolic energy they need. So you need to get blood into your limbs and to your brain. And the process of doing that means that you have to speed up the rate at which the heart is beating, and you have to redistribute the blood in a very precise way. So this is why blood pressure is not a real thing. The rise in blood pressure is not the goal of the fear response. The rise in blood pressure is an artifact of this movement of the blood around in the body to accomplish this goal. Same thing with stress hormones. They aren't in your brain and body to make you feel bad. They're in there to help accomplish certain goals and to keep you focused on a very stressful situation. Requires a lot of body resources, and you may feel worn out and tired and under a lot of pressure when all this is happening. But the point of that is to give you as much energy as you can while the stress and danger are in front of you. So all of these animals undergo fear conditioning. Probably every animal undergoes fear conditioning. It's a kind of universal survival mechanism. You have to learn about novel dangers as well as those that are kind of burned into your brain by evolution. So I already said how we pursue this. We're studying auditory neurons that are transmitting to motor neurons that control these responses. So somewhere along that chain of events between the auditory system and the motor systems are where the plastic changes are that take place. So in every animal that has an amygdala, down the middle line here you see all of the vertebrates basically, from fish and reptiles and birds, various mammals, primates, and so forth. The amygdala is the centerpiece of this kind of learning. So the stimulus, whether it's a light, a tone, an odor, or a general context where something bad is happening, will be routed through the amygdala and will control all these kinds of responses on the basis of plasticity that takes place in large part within the amygdala, but also in other parts of the brain. But we'll be focusing on the amygdala for this evening. Now, if you don't know where it is, imagine an arrow being shot through your eye and through your ear, and where they come together, that's kind of where it is. And it's on both sides of the brain. We say the amygdala, but there's one on each side, like most structures in the brain. So animals like fruit flies and snails don't have an amygdala because they're not vertebrates and they have a different kind of hardware altogether, but they still undergo fear conditioning. So we'll get back to that in a little bit. So how does the amygdala do this? So it's got these inputs from sensory systems. As I said, the auditory and visual systems all converge on the amygdala. And so here's the hiker. This is an innate fear response. So the hiker's walking through the woods and he's about to step on the snake. And his amygdala detects that there's something dangerous there and causes the heart to beat faster and the muscles to tense up and so forth. And there are two ways of getting that information to the amygdala. One is by way of a way station that takes information to the cerebral cortex called the thalamus. And all the sensory systems have this thalamic structure. So the visual system sends its information to the visual thalamus. That sends it to the visual cortex, where you would perceive the snake as an object. But there's a shortcut from the thalamus directly into the amygdala that will allow some crude template to activate the amygdala. And we really have studied this primarily in the auditory system, but when this article was written in Scientific American in 1994, the artist had a much easier time doing it with visual stimuli than with auditory, so that's what he did. Since then, these pathways have been confirmed in the visual system as well. But imagine in terms of an auditory stimulus, so if all of a sudden a bomb goes off in this room, you'll all be freezing and hunkering down, your blood's going to be racing before you really know what's going on. And we think it's through this kind of low road from the thalamus directly into the amygdala that these kinds of automatic responses can be triggered before you actually consciously know what it is you're responding to. So even though both the thalamic and cortical pathways converge on the amygdala, almost all of our work has involved the thalamic pathway because it's a little more manageable experimentally. Now, one of the consequences of this is that when the amygdala is activated by the thalamic pathway, you might not really have the same processing that's going on in the cortex. And one idea from the point of view of psychopathology is that a lot of times we don't understand where our fears and phobias come from, and it may be that in some people, for some reason, their amygdala is being activated by a stick rather than an actual snake all the time. So they are always seeing sticks as snakes, whereas in the normal brain, these two pathways would be coordinated, so the cortex would be able to say it's a stick, not a snake, and so you would stop walking. Here, this is an example. Someone was hiking in the woods somewhere and came across this on the ground and remembered a lecture I'd given, so they sent it to me. But you can see how you could easily mistake that for a snake as you're walking along and all of a sudden you freeze and you realize it's a stick. So the idea is that these two pathways may somehow become dissociated in some people for some reason, so that their amygdala is always seeing snakes where there's only sticks. As I said, Liz Phelps and her colleagues at NYU have been great collaborators, and they've verified that the amygdala is activated in the human brain when people undergo this kind of fear conditioning. If the amygdala is damaged, the fear conditioning doesn't take place. So it's the same as in the rats. In a way, the amygdala, though, is a misnomer because there's not one thing that's really the amygdala. It has a lot of different parts. There's about a dozen parts, and some of them are shown here. And not all of them are required for fear conditioning to take place. So if you lesion different parts of the amygdala, most of them have no effect, so the high bars mean no effect. And you see that there are three areas, the central and the lateral, and the entire amygdala, when damaged, have the effect. So the effects of the entire amygdala destruction there, all amygdala, the fourth column, are replicated by damage to either the central or the lateral nucleus. Now, the central and lateral nucleus is the triangular part of this basolateral region in blue. Central amygdala is the top part of that yellowish region. And so these are, it's important to pinpoint in as much detail as possible the areas that are involved. So we tend to do our work at as fine a level as possible, and we focus almost always on the lateral nucleus to the extent we can. Now, sometimes we hear about the basolateral complex, which is that entire blue thing, but that's in a way like talking about Australia when you really mean Sydney. You need to, there's a lot of territory there that's not included in the part that we think is especially important, the lateral nucleus. So the sensory information comes into the lateral nucleus. The lateral nucleus communicates with the central nucleus both directly and by way of a variety of intra-amygdala connections. And then the central amygdala connects with various areas in the brainstem that control all these different responses that we can measure. Now, the reason that we have to be as precise as possible is because the data take us there. So we started out with the whole brain, we got to the amygdala, we got to the lateral amygdala. Now we're dividing the lateral amygdala in two because the CS and the US, the tone and the shock, converge only in the dorsal half of the lateral amygdala, or primarily in that dorsal half. So the key plasticity, we think, has to be where the tone and the shock come together, because that's the basic principle that underlies the learning. If you're associating two stimuli in the outside world, those stimuli have to meet up somewhere in the brain, and we think the lateral amygdala is where that meeting of the two stimuli comes together. Now, we can record from cells there, and when we do this, we see that there are two kinds of cells in the dorsal half of the lateral amygdala. So if you look on the bottom panel there, you see the LAD where the black cells are, and then below that, the lighter colored representations there. So again, we've divided the amygdala into different nuclei. The lateral nucleus is important. We divided that into two parts, the dorsal and ventral. And now in the dorsal half, we're dividing that into two parts, where you have this very superior part at the apex of the triangle that has cells that learn rapidly and then reset. So that would be the top right. So got 3 trials there. By the 3rd trial, they've learned, and now they reset and back to 0. So they continue, their level of response is back to where it was after they've learned. But the cells below that learn more slowly, and then they hold on to the memory. You can see that the response in the middle panel on the right side remains elevated above the starting point. Now, we think this is important because I told you about people with phobias who have been successfully treated, and so they're not acting afraid anymore, but then the phobia can be brought back. That means that the phobia is living in the brain somehow, but not being expressed. So these cells here, towards the end of the graph there, it says extinction. That's basically cognitive behavioral therapy for rats. The rats are no longer acting afraid behaviorally at this point, but those amygdala cells still remember the danger, and we think those could be a kind of substrate for the reactivation of these fears and phobias later. So we call these trigger cells and storage cells because the trigger cells are involved in the initiation of the plasticity, and the storage cells seem to hold on to the plasticity for a long period of time. So this kind of puts the circuitry all together. We've got inputs coming in, converging on the lateral nucleus from the CS and the US pathways. Lateral amygdala communicates with the central amygdala by way of direct and indirect pathways within the amygdala. Central amygdala controls different areas that are involved in regulating the various kinds of responses. So you could do a similar picture for visual pathways into the amygdala. And the point is that the amygdala these areas of the amygdala are involved in the plasticity regardless of the modality of the input, the CS input, and regardless of the response-output connection. So it doesn't matter what the measure is, and it doesn't matter what the stimulus is at the beginning. The amygdala is the kind of universal sensory motor independent part of the circuitry, and that's why we focus so much on what's going on in there. Now, so again, the CS and US are converging in the dorsal half of the lateral amygdala, so we do a lot of our research there. And sometimes we actually take the amygdala out of the brain and study it as a brain slice living in a dish. And this allows us to study the molecular mechanisms underlying plasticity in much more detail. And then we can compare that with results that we get when we go back to the whole animal and perform the same kinds of molecular manipulations, looking at the effects on actual learning in the behaving rat. So by going between these kinds of procedures, we and others have made a lot of progress in working out a lot of the molecular machinery that's involved. What's interesting is I'm not going to go through all the molecular details here, but what's interesting is that the molecules involved in fear conditioning in the rat and in invertebrates like the aplysia are very similar. So this goes back to the issue I raised earlier, which is there's no amygdala in an aplysia, but the molecules that are involved are very similar. So there's a kind of conservation of the molecular machinery for the learning in these widely divergent kinds of animals, both sea slugs and bees and flies. This is all highly conserved. But it's also true in other parts of the mammalian brain. So in rats and mice, areas like the hippocampus, which are involved in our conscious, explicit cognitive memories, also use very similar kinds of molecules. So what this means is that the difference between a cognitive memory formed by the hippocampus, which allows our memories to be consciously accessible, and an amygdala-based memory, which is an implicit form of learning that is not consciously accessible directly within the brain. They use the same kinds of molecular machinery. So while someday we may discover molecules that distinguish hippocampal memories and amygdala memories, I think at this point the similarities outweigh the differences, and what we have to conclude is that for the mammalian brain, the difference between a cognitive memory and an emotional memory like this is not the molecules, but the circuitry in which the molecules are acting. So I've said circuits weren't important between, it's not necessary to have the circuitry to have this kind of learning. But when you have the circuitry in the brain, you see the different circuits are performing different kinds of functions. So hippocampus its inputs and outputs are largely with other cortical areas so that you can have more and more elaborate processing of the same stimulus. Whereas with the amygdala, the connectivity is with outputs. So when the amygdala is active, you get these automatic outputs that allow the responses to be expressed. Now, these are gross oversimplifications of what's going on in the hippocampus and amygdala, but I think you see an important difference between the way a fear memory is expressed by way of amygdala outputs and how the memory about a fearful situation might be regulated by the hippocampus. So we might think of these as memories about emotion as opposed to emotional memories. So the amygdala would be involved in the emotional memory, implicit, unconscious, response related, whereas the hippocampus might be more involved in memory about emotion, which is facts and experiences about particular emotions that you might have had. Let me now move to some areas of more recent research, and here we'll get a little more technical in terms of some of the details. But the lab is working on 5 or so major projects. One is just trying to understand more and more about the molecular mechanisms underlying the way fears are acquired and stored. Another is the relation of synaptic plasticity, especially so-called Hebbian plasticity, to fear conditioning. We also are doing a lot of work on individual differences. A lot of research in general neuroscience is a science of the ordinary, but if we're interested in pathology, we can't deal with the typical situation. We have to understand the extremes of the responses. So we're trying to get more and more into these individual differences. We've done a lot of work on the lability of memory after the retrieval of memory. That's called reconsolidation. And also on how the brain transitions from fear reactions to actions. And I can't really talk about all of these tonight, so I'll just briefly talk about three of them, starting with the molecular mechanisms and some work we've been doing on the contribution of norepinephrine to fear conditioning. Now, if you know anything about norepinephrine in learning, its general role is thought to be one of consolidating memories. So something happens, and then after that period, the memory goes through a phase of short-term memory, which lasts for a little while, in the case of these kinds of memories, a couple of hours. And then it's consolidated into a long-term memory, and these are different processes. Now, norepinephrine's role is generally thought to be that of consolidating or converting the short-term memory into a persistent long-term memory. But we have a new evidence. This is never, in spite of the importance of norepinephrine to fear and anxiety in people, the relationship of norepinephrine to fear conditioning has not been thoroughly studied. There have been some studies here and there over the years, but it hasn't been thoroughly investigated. So we decided to revisit this. And so what you see here is on the top where it says A, the rats are trained, they undergo fear conditioning. And in panel A down below, what you see is that if you inject the drug, norepinephrine, in this case a norepinephrine blocker or beta-adrenergic blocker, propranolol, into the lateral amygdala prior to training, you disrupt short-term memory and long-term memory. But if you inject it immediately after training, which is the way you usually do these consolidation experiments to show that short-term memory is intact and long-term memory is impaired, what you see is there's no effect on long-term memory at all, and short-term memory is unaffected as well. I don't have it shown here, but it's not affected. So the point is that norepinephrine blockers, beta-adrenergic blockers, put into the lateral amygdala, disrupt something before learning, but they don't disrupt anything after learning. So they're not affecting the consolidation of the memory in this situation. What they're doing is altering the way the learning takes place. And this is a very, very different view from the one that's usually talked about in terms of the role of norepinephrine in memory. And you can do the same thing if you give the noradrenergic agonist, isoproterenol, into the lateral amygdala. You get a boosting of learning if you do it before training, but no effect if you do it after training. Top is before, bottom is after training. So neither the blockade of norepinephrine receptors, beta-adrenergic receptors in the lateral amygdala, nor the stimulation of those receptors after training is having any effect. The only effect is occurring before training. Now, what's interesting here is that you can replicate this effect by giving a single shock or some other stress prior to training. So the idea is that the shock is releasing the norepinephrine from the locus ceruleus, which it's a part of the midbrain that has fibers going to the amygdala and the rest of the forebrain. So the shock is activating the locus ceruleus, and that's releasing the norepinephrine in the amygdala, and that's facilitating the learning. But the shock doesn't occur until the end of the first trial. So if you do this with one trial learning, you have no effect of norepinephrine blockade on fear conditioning. But if you do it with 2 or 3 trial training, you get the effect. So again, this shows that I don't have the slide, but this shows that the the shock on the first trial where you get a pairing causes the release of the norepinephrine, and on the second and third trial, the norepinephrine is now floating around and able to facilitate the learning. So this raises the questions about exactly what's going on in terms of learning. And the usual idea that we have in neuroscience about learning is that it's a Hebbian process, which is named after the psychologist Donald Hebb, who said that when you have a strong stimulus that activates a cell, and if the cell is receiving a weak stimulus at the same time, the weak acquires some of the benefit of the strong, so that the weak can then produce the same effect. So we decided to test this using new techniques that are now available, a technique called optogenetics. And I don't have time to explain all the gory details, but basically what you do is you put a light-sensitive molecule into the amygdala by coupling it to a virus, and you inject the virus with that molecule into the amygdala. The virus then takes it into the amygdala cells, and then you let the animal recover for a while, and then you put a little fiber optic cable, very tiny cable, into the amygdala and give a light flash. And each of the, on the top right there, each of the lines up there is a set of action potentials that are elicited by that light flash. Now, we obviously didn't invent this technique, but Carl Diceroff is the mastermind of this wonderful approach. But so each time you flash the light, you get a very precise millisecond by millisecond activation of those cells. Then if you stain the brain afterwards to see where the activity was, on the side that was stimulated with the light, you see a lot of activity in the lateral amygdala. On the other side, there's nothing. So this is just kind of a proof of principle. So what we wanted to do was test the Hebbian hypothesis using these light-activated responses as the strong stimulus. So we're causing the cells to fire action potentials each time we flash the light. So we wanted to pair this on a millisecond by millisecond level with brief tone pips. So we give a tone pip. This is an external real stimulus in the outside world, and that's going to meet up in the lateral amygdala with this millisecond level activation of these cells. And what it does, just go to the bottom line, is it creates learning. So we create fear conditioning by simply turning on the amygdala cells millisecond by millisecond at the same time that they're getting a tone pip. And the controls don't show this learning. So if you look on at the magnitude of the learning, though, on the right side here, in the long-term memory test, it's a little below 20% or so. That's not a lot of freezing. I mean, it's highly significant, but it's relatively weak learning. So we wanted to explore the possibility. I tell you that norepinephrine is boosting learning. We wanted to see if we can boost learning by combining these experiments with optogenetics with norepinephrine manipulations. So the first thing we did was to ask whether we can potentiate weak conditioning. So what we want to do is do weak fear conditioning with a tone and a shock, real conditioning, and that's what we have here. By instead of giving like the normal conditioning up there that we used in the optogenetic thing, would be similar to like giving 16. I mean, we're titrating this a bit. So basically, you've got a strong protocol and a weak protocol, and the weak protocol is going to be used to see if we can alter this with the by combining the laser stimulation with the weak conditioning. And so in the middle panel is the weak conditioning. If you then give the laser stimulation alone, you boost it up a bit. So we can boost normal conditioning by depolarizing the cells. Now the question is, can we alter this with norepinephrine manipulations? So here we have the weak CS plus the laser stimulation, weak shock, tone shock pairing plus the laser stimulation in the vehicle group there. And if we give the drug propranolol, which blocks the norepinephrine receptors in the amygdala, we reduce that effect. So we reduce the boosting effect that we got by giving propranolol. And we can then do something else, which is to combine isoproterenol and the laser stimulation. And that alone is enough to create some conditioning. So all of these things point to various ways in which depolarization of amygdala cells combined with tones can be altered by giving norepinephrine manipulations. So we think we're getting at a way that norepinephrine is working that's somewhat different from the way it's been viewed in the field of behavioral neuroscience as a memory consolidator. Instead, what we're doing is looking at the role of norepinephrine as a learning molecule, a molecule that is going to facilitate the learning process. Okay, reaction versus action. This is going to be the topic of my presentation tomorrow at the FEAR symposium that Bernard Baleen has organized. But I'll give you, for those of you who aren't going to be there, I'll have a brief presentation of it here. Now, so this is 2008. In October of 2008, the stock market is crashing in the morning, and so he's watching it happen. So he's freezing, basically, and he's going to run up and change his holding. So he's going to transition from his fear reaction to an action that he hopes will relieve some of the aversion that he's experiencing here. So how do we model that in the laboratory? We use something called active avoidance. So here the rat's in a chamber, and it gets a shock when the tone is presented, and what he learns is that if he hears that when the tone comes on, if he runs over to the other side, he doesn't get the shock. So he's actively avoiding the shock by using the tone as a warning signal. So what you can see is that the learning of the active avoidance response on the top is the inverse of the loss of the freezing response. So obviously a rat that's getting shocks in this box when there are tones and so forth is going to first having a fear response because he doesn't know what's going on. So the tone is predicting the shock, and so he's going to freeze to the shock. And the shock is also conditioning the chamber itself as a fear cue. So he's doing a lot of freezing. But then you see that as the freezing response goes away, the active avoidance response is acquired. Now, this makes sense because you can't take action if you're frozen, right? They're kind of counter forces. You can't do one if you're doing the other. And I'll come back to that in a minute. So what's interesting is that the parts of the amygdala involved are both similar and different from what I've told you about so far. Our friend, the central nucleus, is not involved. It's the line on the far right there, but the lateral and the basal amygdala are involved. So this gives us a somewhat different picture from what we've been seeing, where the CS and US have to converge there in the lateral nucleus, and then the lateral communicates with the central nucleus for the control of these fear reactions, but then with the basal nucleus for the control of the fear actions. But what I want to just sort of close out with is the idea of the individual differences in avoidance. So about 20 or 30% of the animals that go through an avoidance experiment don't learn at all. And we wanted to, usually these are thrown out, but we wanted to know why they weren't learning. So we divide them into these are summed across a lot of experiments here, but what you can see is you have animals that are good performers on top in the red, it's kind of the average up there, and then the poor performers in blue at the bottom. So they just don't learn to perform the avoidance response. And the question is why? And well, one of the things that happens is that in the good performers, the freezing response goes away, but in the bad performers, or the poor performers, they not only continue to freeze, but increase their freezing a bit. So the question is, what would happen if we lesion the central amygdala, which controls these freezing responses? And what you see is that on the left side, these are the poor performers. They're just not learning the response. And so we said, well, maybe when we lesion the central amygdala, they'd be able to learn. But to our surprise, not only had they learned, not only could they learn, but they had already learned. On the first day they're tested after the lesion, they already show significant avoidance responses, and they learn it a bit better. But what this means is that they were so frozen in fear that they couldn't do the adaptive thing that would allow them to not get shocked. They just got stuck there. And again, this sounds like psychopathology in a way. So, that's interesting, but we can't go in and lesion the central amygdala of people who are just frozen in fear. So we go back to our propranolol data, and we gave propranolol systemically to rats. Systemic means just give them a shot of it, the way you would pop a pill, and you can see that the animals are less fearful. And this is just a replication of studies that a number of people have done over the years. So propranolol is known to reduce fear responses. It's often used as a treatment for stage fright. People take a little propranolol before they do public speaking or perform in various ways. So this all makes sense, but then the question is, what does it do for these poor performers? And what you can see is that the poor performers with propranolol, as the sessions get longer and longer, get better and better and actually achieve the same level of performance as the good performers when they have propranolol. So I know that this is already in use to some extent in the treatment of fear, but this is further encouragement that this might be an important thing to use in the effort to kind of get past these passive coping strategies into more adaptive, active coping. And I think in the interest of time, I'll skip some of these gory details and just close with the question of, what is an emotion and what do freezing rats tell us about emotion? Well, as I said at the beginning, I don't think they're telling us much about human feelings. The feeling of fear is fear is a word. It's an English word, obviously. If you look it up in the dictionary, you can find, I think, 37 or so words related to fear and anxiety that all kind of circle around the concept. And so if you're a person with a lexicon like that, you can assign all kinds of variations of fear and anxiety by the word you choose to label your state. A rat or other animal is unable to draw upon that kind of linguistic sophistication to cognitively interpret the situation that it's in and partition it in those kinds of ways. So, I'm sure the rat is going through some kind of experience, but it's not going to be anything like what a human experiences. As the philosopher Thomas Nagel said in his famous paper, "What is it like to be a bat?" Well, it's like being a bat. So it's not something that we can, we don't have sonar, so we can't appreciate what the bat's sensory world is like, or its emotional world either. So I just want to emphasize that when we do these studies, for me, I'm more comfortable talking about the way the brain detects and responds to danger. And I think I've also shown you how this research is relevant to humans, because we can devise strategies that help us understand not only ways that people experience and deal with fear, but also better ways to perhaps treat certain fear disorders. So thank you very much.